Hello and welcome to the HIV-RNA-TestGuide.com YouTube channel, your online partner for quick, affordable, and 100% confidential HIV testing. Whether you're looking for quick, confidential results or simply want to stay informed about the latest in HIV research, you're in the right place. For more information, visit HIV-RNA-TestGuide.com. Check the link in the description below or visit the bio section of our channel. You can access our more than 4,500 HIV testing labs across the United States, just a click away. So, without further delay, let's start today's video.
Have you ever thought about what a truly HIV-free future would actually look like?
It's a powerful thought.
For decades, that whole concept has sounded less like a medical goal and more like something, pulled straight out of science fiction.
Yeah, a destination a lot of people marked as, frankly, scientifically impossible.
But today, we are talking about a breakthrough that is changing that roadmap completely. And this isn't just some theoretical hope. We're talking about a highly engineered biological solution.
What's so fascinating here is that the scientific community is, it's moving past the decades-long strategy of just managing the virus, of just containment.
Right.
We are now pivoting to engineering the human body to achieve long-term self-sustained remission. And this pivot, it isn't just a small step. It's a foundational change in the battle plan.
Okay, let's unpack this. Welcome to the deep dive. Today, we're focusing on a cutting-edge immune-based therapy, and it has a very sci-fi name. IMC-M113V.
It does.
For you, the listener, this is so much more than just a new drug. This therapy, it belongs to a whole new generation of treatments designed for really ambitious goals. Eliminating the hidden virus, completely restoring immune function, and this is the big one, achieving long-term remission without needing daily medication for life.
Exactly. And our mission today is to synthesize the really complex, personalized science behind this T-cell therapy into clear, comprehensive knowledge you can use. This approach, it just fundamentally shifts the battle from generalized suppression.
Which depends entirely on someone taking a pill every single day.
Every single day, without fail. It shifts from that to direct, targeted destruction of the very cells where HIV has traditionally hidden out. So we're going to cover exactly what this revolutionary T-cell receptor therapy, or TCR therapy, is.
We'll get into the science of how they actually program these T-cells to become precision hunters.
And we'll talk about why this specific development holds such crucial promise for the U.S. population right now, and what that ambitious 10-year roadmap might look like for achieving, that ultimate HIV-free future.
And crucially, we have to talk about the paradox here. As the science gets more futuristic, the basics, they actually become more essential. We'll be connecting this revolutionary engineering right back to the absolute foundation of public health success. How advanced HIV testing is still the non-negotiable tool for making any of this possible.
Couldn't be more important.
So, get ready, because we have a lot to dig into. Okay, let's start at the very beginning. If I'm a listener and I hear IMC-M113V and T-cell receptor therapy, my first thought is probably, that's a major league science word salad.
Chuckles. It is a mouthful.
So how do we define this therapy simply? And maybe more importantly, why is it such a necessary step beyond what we already have, which is, our current gold standard?
That's the perfect place to start. Let's define it by contrast. Think of IMC-M113V as engineering your own immune system with upgraded, highly specialized, and extremely persistent precision tools.
Okay, so it's a software upgrade for your cells.
In a way, yes. It's a T-cell receptor therapy, or PCR therapy. And we absolutely have to contrast it immediately with antiretroviral therapy, or ART, which has been the cornerstone of HIV treatment for decades.
And let's be fair, ART is one of the great medical triumphs of our time. It allows people with HIV to live full, healthy lives, often with undetectable viral loads, which means the virus can't be transmitted.
A miracle, truly.
But that leads to the critical question the listener is probably asking right now. If ART already gives people long, healthy lives, why would we pivot to this incredibly expensive, personalized, genetically engineered therapy? What's the major risk-reward calculation we're missing here?
That is exactly the right question to start with. The limitation of ART. It lies in its mechanism, how it works. ART suppresses the virus globally in the body by just interrupting its life cycle, stopping it from making copies of itself.
So it basically puts the virus to sleep.
It's like putting the entire viral population into an induced coma. But, and this is the critical but, it cannot, by its very design, physically eliminate the virus that has already woven its genetic material into the host's resting immune cells.
So it maintains the ceasefire, but the enemy has these deep bunkers hidden underground.
That is a perfect analogy. The virus establishes these latent hideouts, these viral reservoirs, in specific types of immune cells, particularly memory T cells, and in tissues like the lymph nodes and the gut.
And because those cells aren't actively making more virus.
RRT drugs, which target the replication machinery, can't affect them. The drugs just pass right by. That latency, that hiding, is exactly why people have to take RT every single day for the rest of their lives, because if they stop, even for a short time,
The virus wakes up.
it reemerges from those reservoirs and the viral load just comes roaring back.
And that's where IMC-M113V changes the entire game. It stops trying to suppress the replication and instead teaches the body's own immune system to find and destroy the bunkers themselves.
Exactly. It arms your existing T cells, your body's defense force, with brand new, specific intelligence. This engineered intelligence allows the T cells to function like these specialized, personalized search and destroy units.
But they're not just general soldiers anymore, they're special ops.
Precisely. They seek out and destroy HIV-infected cells directly, and crucially, that includes the ones hiding inside the viral reservoirs. This shift is it's profound. We're going from maintenance to elimination.
Okay, let's drill down into that specificity because the source material really focuses on the exact target. It's the HLA A02.01 restricted GAG peptide. It sounds like highly specific jargon.
It is. It is precise, but precision is the key to defeating a virus as shifty as HIV. Let's break that down into 3 parts.
Okay.
First, the HLA A02.01. Every cell in your body has these HLA markers, human leukocyte antigens, on its surface. They act like personalized ID badges.
Like license plate for the cell.
Exactly. And this specific type, A02.01, is one of the most common HLA types in the world. It's found in nearly a third of the US population, which makes this therapy immediately relevant to millions of potential patients.
Okay, so the HLA A02.01 is the specific model of the ID badge this T cell is programmed to look for. What about the GAG peptide part?
So when a cell gets infected by a virus, it processes the internal viral proteins. It basically chops up little pieces of the virus, these are the peptides, and it displays those pieces on the surface using that HLA molecule, that ID badge.
It's like the cell is raising a little flag saying, something's wrong inside me.
That's exactly it. And the GAG peptide is a piece of a core structural protein of the HIV virus. It's relatively stable, meaning it doesn't mutate as fast as some other parts of the virus, and this is key. It's expressed even when the virus is in its latent sleeping state.
So the engineered T-cell receptor is custom built to recognize only the HLA A02.01 badge when it's carrying that specific GAG peptide flags, like a double security check.
That is the entire mechanism. The TCR therapy is engineered to see that precise telltale combination. And the genius of this approach is that GAG is present inside the infected cell, even if the virus is hiding, giving the T-cell new eyes to spot an infection that standard drugs just can't see.
We mentioned why we can't just target the virus on the outside, but let's just reiterate that point because it really underscores how new this is. Why is seeing inside the cell so necessary when the virus is playing dead?
It's all about minimizing the visible footprint. When HIV is actively replicating, it puts a lot of viral proteins on the outside of the cell. That's relatively easy for a normal immune system to spot.
But in the reservoir?
In the reservoir, in those resting memory T cells, it's producing very little, sometimes almost no, external viral proteins. It's basically cloaked. But the cell is still a biological machine, right? It's still processing some internal components like that GAG peptide, and it's still displaying them on the surface via the HLA complex.
So even if the virus isn't shouting, the cell itself is whispering. It gives off this faint internal residue that the engineered T cell can detect.
That's a great way to put it. This therapy is programmed to recognize that whisper, that faint signal. This allows it to identify hidden HIV even when the viral load is undetectable by standard tests, when the virus is essentially playing dead. IMC-M113V aims to remove this ability to hide entirely.
Which is the one thing RT just cannot do.
Exactly. It's changing the operational definition of what undetectable can truly mean.
So we are not just teaching the immune system to fight the war, we are giving it a molecular target acquisition system to find the absolute deepest, quietest bunkers. This is the pathway then to making a functional cure where the body manages the virus on its own without medication. A viable scientific goal.
That's the goal. A true functional cure.
Okay, here's where it gets really interesting for me. This therapy is highly personalized. It involves what sounds like factory-level biological engineering. It feels like we are talking about building a custom, upgraded army, one person at a time.
That's not far off.
The source material breaks down the mechanism, into 5 pretty manageable steps. I want to emphasize how new this is, but also I have to ask about the risks involved.
That's crucial. The novelty here lies in adapting these highly sophisticated techniques from oncology, specifically CAR-RT and related T-cell engineering, to the completely unique challenge of a chronic viral infection. Cancer is often a bulk removal problem.
You find the tumor, you remove the tumor.
Right, but HIV is a deeply hidden latency problem. It's a different beast entirely.
We should probably pause for a second on that CAR-T connection, because while it's similar, IMC-M113V is a TCR therapy, not a CAR-T therapy. What is the subtle, but I imagine vital difference between those two approaches?
It's a really important distinction for the listener. CAR T cells typically recognize targets proteins that are sitting on the surface of a cancer cell. They don't need that HLA ID badge to see their target.
They just see the enemy flag directly.
Right. But TCR therapy, like IMC-M113V, is designed to recognize those internal viral peptides that are presented by the HLA molecule. It needs the ID badge and the flag. This makes TCR therapy ideal for tackling the viral reservoir because it allows the T cell to see those deep, hidden infections that are only broadcasting that tiny internal signal via the HLA complex.
So it's a more specialized tool for a more specialized hidden problem.
It's an adapted, highly specialized platform for latency. Exactly.
Okay, got it. Let's look at the engineering process step by step.
Yeah.
How does this actually begin for a patient?
So, step 1, extracting T cells. Doctors take a sample of the patient's own immune cells, specifically their T cells, through a procedure that's pretty similar to donating blood plasma.
This is personalized medicine at its most fundamental level. We're using your own cells.
That's right. The procedure is called apheresis. It's a detailed, multi-hour process where blood is drawn. Specific components, in this case, the white blood cells that contain the T cells, are separated out, and the rest of the blood is returned to the patient.
So you're harvesting the body's existing defenders.
Yes, ensuring the foundation of the therapy is autologous derived from the patient themselves. This minimizes the risk of rejection. We're just taking their cells out to give them their mission upgrade.
Which brings us to step 2, reprogramming them. These cells that were taken out are then genetically engineered with that specialized receptor that lets them identify the HLA A02.01 GAGA peptide combination.
This is where the synthetic biology takes over. In a very controlled lab environment, scientists use what's called a viral vector, often a modified harmless lentivirus, to deliver the genetic instructions for the new, specialized T-cell receptor right into the patient's T-cells.
So you're using a disarmed virus to deliver the software update.
Precisely. This genetic payload rewrites a part of the T-cell's identity, equipping it with the radar to spot the hidden virus. This is the highly specialized proprietary part of the whole process.
Now, I have to ask the skeptical question. When you're introducing new genetic material, even with a supposedly safe viral vector, what are the primary safety concerns? Could this engineered cell accidentally start targeting healthy tissues?
That is the paramount safety concern in all T-cell engineering. It's called off-target recognition, where the engineered T-cell might attack healthy cells that just happen to express a protein that looks a little bit like the GAG peptide.
A case of mistaken identity.
A very dangerous case of mistaken identity. Researchers mitigate this risk by designing the TCR to be extremely high affinity and very, very specific to that exact HLA-GAG combination. And the clinical trials, they focus intensely on monitoring for any signs of autoimmune reactions or other issues that would signal the T cells are attacking the wrong targets. It's why this therapy is only given in highly specialized centers.
That makes the next step even more crucial. Step 3, multiplying them. You don't just need one of these engineered cells to fight the virus. You need a whole army.
A massive army. Millions, sometimes billions, of these newly upgraded T cells are grown and amplified in a specialized lab.
How hard is that to do?
This in vitro expansion phase is technically demanding. T cells are complex, fussy cells. Growing them to the necessary therapeutic scale requires specialized bioreactors, precise nutrient mixtures, and regulatory signals to make sure they stay potent and viable.
So you have to make sure they're not just numerous, but also still effective.
Exactly. If they multiply too much, they might exhaust themselves. If they don't multiply enough, the infusion won't be strong enough to tackle the reservoirs spread all throughout the body. You need that critical mass of highly specific hunters to be effective.
Okay, so you have the army. Now, step 4, infusing them back. The engineered, amplified cells are then prepared and put back into the patient's bloodstream, usually through an IV. This is the moment the therapy officially begins.
And this step often requires the patient to undergo a brief, low-dose chemotherapy regimen beforehand. It's a process called lymphodepletion.
Why is that necessary?
It temporarily reduces the number of the patient's native unengineered T cells. It essentially makes space for the new engineered cells to move in, set up shop, and establish dominance in the immune system. This pretreatment gives the infused cells the best possible chance to survive and start their patrol.
And finally, step 5, tracking viral reservoirs. Once they're back in the body, these modified cells immediately begin hunting down, detecting, and destroying those HIV reservoirs, the hidden areas that have made standard art insufficient for a cure.
The term tracking is key here. The whole process is a sophisticated immunological search and destroy mission. And the functional advantage is clear. This is the first time we've seen a therapy with this triple functionality.
Okay, what's the trifecta?
First, it seeks HIV-infected cells using those specific HLA markers. Second, it attacks the hidden virus, the reservoirs, which addresses the Achilles heel of ART. And third, it works independently of daily ART because the T cells are now a living, self-sustaining part of the patient's immune system, providing durable, long-term surveillance.
That independence, the fact that this is a living therapy that patrols long after the infusion, that's what moves us from suppression toward potential elimination. The T cells are designed to stick around.
They are. The goal is long-term immune memory and surveillance. If this is successful, IMC-M113V supports the move from simple, chemically driven viral suppression toward true biological HIV elimination and long-term remission, potentially freeing the patient from that lifelong dependence on drugs. That is the ambition.
Okay, so we've established that the reason RT demands lifelong dependency is that it can't clear out those hidden sleeper agents, the viral reservoirs.
And that dependency, it's not just a medical inconvenience, it's a massive logistical, economic, and psychological burden for people.
Right, lifelong adherence to any medication is difficult, and the consequences of stopping ART, even for a little while, can be swift. Viral rebound, increased risk of transmission, and the possibility of developing drug resistance.
The functional cure lifts that burden entirely.
So let's define exactly what a functional cure means in this new context, because I think it's different from what people might imagine. How does it differ from say, a sterilizing cure?
That's a great distinction to make. A sterilizing cure is the absolute total elimination of every last bit of replication-competent HIV from the body. This has only been achieved in a tiny handful of cases worldwide, usually after really complex, risky procedures like bone marrow transplants for someone who also has cancer.
So not a scalable solution.
Not at all. It's highly invasive and not something you could offer to millions of people.
So if a sterilizing cure is total eradication, what's the achievable goal? What is the functional cure?
A functional cure means the virus is still technically present in minute, non-harmful amounts within the body. You might find a few hidden genomes that the immune system just can't access. But the key is that the patient's enhanced immune system controls it completely without any need for daily medication.
So what does that look like for the person?
For the person, it means they are symptom-free. They have no progression to AIDS, they take no daily medications, and their viral load remains undetectable even when they're off treatment. Essentially, their own body has become the permanent ART.
And the mechanism for achieving that, the target that has to be cleared, is that viral reservoir? The hope is that IMC-M113V clears out enough of those reservoirs so that the person's remaining immune system can handle any tiny traces left behind, preventing a rebound.
Precisely. You have to reduce the size and the competence of that reservoir below a critical threshold. You need to eliminate what we call the seed competence, the ability of those latent viruses to suddenly reactivate and flood the system if RT stops. That's the job of these engineered T cells.
So is this all just theory at this point, or are we seeing real-world confirmation that this is actually possible? What does the early clinical data tell us so far about IMC-M113V?
Well, the early data from the phase I&II trials, while it's still preliminary and mostly focused on safety and feasibility, it provides some highly encouraging indicators. The scientific community is using this term high-impact cure pathway for a reason.
Okay, let's get into those specific promising signs. First, they reported strong targeting ability. What exactly does that confirm?
It confirms the core thesis of the engineering. In lab models and in the initial human participants, researchers were able to verify that the engineered T cells successfully navigated the body and located the infected cells that were displaying that HLA-A02.01 GAG peptide complex.
So it's the ultimate proof of concept. The soldiers found their precise, intended target.
It validates the highly specific molecular lock and key design. They went where they were supposed to go.
Okay, second, and this is vital for any future expansion, is the promising safety profile. You mentioned earlier the severe risks with some T-cell therapies in oncology. What's the data showing for HIV patients?
Safety is paramount, right? You're introducing genetically modified, highly active cells into a person. Some of the first-generation cell therapies, particularly CRT, sometimes triggered this severe, even life-threatening systemic inflammation called cytokine release syndrome, or CRS.
And we're not seeing that here.
The source material really emphasizes that participants in the early IMC-M113V trials tolerated the therapy well. They showed only mild or manageable adverse events, which, in the world of cell therapy, is a massive clinical success.
So a good safety profile means this therapy can move forward into bigger efficacy trials with much more confidence.
Absolutely. Early tolerability is the green light. But the third point, this is the real headline for the cure agenda: potential reservoir impact.
Now, how do researchers even measure the shrinking of a reservoir that's specifically designed to be hidden from standard detection?
It's not easy. They use these highly specialized, laborious, and expensive assays. These are not your standard clinical tests. For example, they do something called a quantitative viral outgrowth assay, or QVOA.
What does that involve?
They take blood and tissue samples from the patient, and they activate the resting T cells in vitro in a dish to see if they cough up any replication-competent virus. If the IMC-M113V therapy is working, the number of cells that are capable of reactivating the virus, the size of the reservoir, should measurably decrease over time compared to where they started.
And that's what they saw.
The early data showed exactly this: signs that the reservoir size was shrinking.
That is the definitive metric. RT can't shrink the reservoir, it just stops the virus from leaving it. IMC-M113V is demonstrably dismantling the hiding spots.
Correct. Seeing a measurable reduction in the reservoirs suggests that the therapy is doing exactly what it was designed to do: hunting down and killing those resting infected cells. And this leads to the fourth indicator: possible long-term control.
Meaning the cells stick around.
Right. Early responses suggest that the engineered T cells are not just doing a one-time cleanup. They appear to be establishing a sustained immune presence, maintaining surveillance over many months. This persistence is absolutely key for a durable functional cure.
So the scientific consensus, then, is that this isn't just a marginal improvement. This is a potential paradigm shift.
That's the so what. When you hear U.S. researchers labeling this specific T-cell pathway a high-impact HIV cure pathway, that's significant. It means they believe they've validated a mechanism that appears capable of solving the central, enduring challenge of HIV latency and the reservoir. It represents the best biological route we have toward moving from daily management to genuine biological control.
We have to move this conversation from the complexity of the lab to the reality of public health. We're focusing on the U.S. population, where HIV remains a critical, ongoing national health issue. It affects over 1.2 million people.
And that scale is exactly why this isn't just some academic curiosity. While the U.S. has excellent treatment programs and high access to RT compared to many parts of the world, the cost and the logistical complexity of managing this for life are still enormous societal burdens.
Burdens that IMC-M113V could potentially alleviate.
Potentially, yes, entirely.
So let's detail the economic and systemic reasons why U.S. scientists are so profoundly excited about this therapy's potential, specifically for the national population.
You have to start with the economics. The average lifetime cost of treating just one person with HIV in the U.S. is estimated to be over $400,000.
Wow.
And that's driven largely by the high cost of decades of continuous RT medication, regular monitoring, managing other health issues. It all adds up.
But a T-cell therapy like this will initially be extremely expensive, right? We're talking 6 figures for a one-time treatment.
Absolutely. But if you can eliminate the need for 50 years of daily medication, you generate massive long-term savings. For individuals, for insurers, for the entire healthcare system, the potential return on investment is generational.
So the economic benefit is huge, but what about the patient-level benefits? The things that RT, for all its effectiveness, still can't address.
First and foremost, the potential to reduce lifelong medication dependence. Adherence is a massive challenge for any chronic disease. Missing doses can lead to a detectable viral load, potential transmission, and the development of drug-resistant viral strains.
If this is successful, it literally takes millions of people off that compliance treadmill.
It does. And second, it helps the body regain innate, permanent control over the virus. This is a biological fix, not a chemical one. It removes the dependency on continuous chemistry to stay healthy.
And from a public health policy perspective, this aligns perfectly with current U.S. objectives.
Absolutely. This development directly supports national HIV reduction campaigns, specifically the Ending the HIV Epidemic in the U.S. initiative.
How so?
Well, that initiative has 4 pillars: diagnose, treat, prevent, and respond. A functional cure tool that reduces transmission risk and eliminates treatment fatigue is the ultimate fulfillment of that treat pillar. It allows the body to maintain viral suppression even when a patient might face challenges with adherence.
We also have to acknowledge the equity implications here. Health crises rarely affect all communities equally. The sources really emphasize that the communities with the highest HIV impact stand to benefit the most.
This is perhaps the most profound potential benefit of all. The source material highlights that the African American, Latino, southern states, and urban populations have consistently borne the heaviest burden of the HIV epidemic in the U.S. in terms of both incidence and mortality.
And that's due to historical inequities, barriers to consistent access to care, and just concentrated epidemics.
All of the above.
But if the root issue is access and adherence challenges, how does a highly personalized complex T-cell therapy solve that? Wouldn't it create more access challenges at first?
It's a complex trade-off, you're right. But the long-term benefit is overwhelming. While initial access to the therapy will be centralized, removing the requirement for daily, lifelong adherence solves the core, persistent problem of treatment compliance.
I see. So it's a one-time, high-intensity intervention that removes a lifetime of lower-intensity barriers.
Exactly. For communities that are struggling with socioeconomic barriers, housing insecurity, food insecurity, or just inconsistent healthcare access, the pressure to obtain, store, and take pills reliably every day for 50 years is immense. Removing that requirement is a monumental step toward closing health equity gaps and stabilizing public health outcomes in these critical populations. A functional cure, once you can scale it, is a great equalizer.
Let's just take a moment to imagine the potential social impact here. If IMC-M113V proves successful and becomes scalable, what does America look like in a decade?
The transformation would be hard to overstate. Imagine an America where hundreds of thousands of people who were previously relying on complex, lifelong medication regimens no longer need them. The fear of viral rebound just dissipates. HIV transmission plummets because the overall viral presence in the population is dramatically reduced, and the reservoirs are managed biologically.
And the mental health impact, the decades of fear and the burden of stigma that has surrounded HIV that would begin to collapse as the disease transforms.
Yes. When a disease shifts its status from a chronic, deadly, and stigmatized condition to one that's biologically managed or functionally cured, the social stigma loses its power. Testing becomes normalized. It's viewed not as a life sentence or a permanent medical identity, but as a simple, proactive step toward accessing a curative solution. IMC-M113V, therefore, isn't just a biological breakthrough. It represents hope backed by science, capable of fundamentally restructuring the social and psychological landscape of the disease. But this raises a really important question that requires us to ground this futuristic discussion. We've been talking about this incredible personalized therapy that involves genetic engineering, apheresis, armies of T cells.
Right.
Even with breakthrough treatments like IMC-M113V on the horizon, why is HIV testing the most basic tool we have, still the non-negotiable core foundation of prevention, treatment, and even eligibility for these new cures?
It seems counterintuitive, doesn't it? Why should I care about my test status today if the cure is 10 years away? It feels like we are asking people to focus on the horse and buggy when we're discussing a spaceship.
Because you cannot engage the spaceship without the data provided by the horse and buggy. You cannot start a precision immune therapy without knowing, with absolute certainty, the status of the patient's virus, the health of their T cells, and the specific genetic markers that determine if they're even eligible.
So testing provides the necessary baseline and all the ongoing monitoring data at every single stage.
Every stage. Let's look at the essential role of advanced testing today, starting with the most basic function: early identification.
Okay.
Early identification is fundamental because it dramatically impacts your treatment options down the line. Advanced tests, specifically fourth-generation antigen antibody tests, or the even faster HIV RNA tests, can diagnose an infection sometimes as early as 10 to 14 days after exposure.
And for a therapy like IMC-M113V, getting that early diagnosis is vital.
It's vital. The earlier a person gets into care, the healthier and more intact their T-cell population will be.
And why are healthy T cells so important for a T cell therapy? It's like the answer is obvious, but let's spell it out.
Well, IMC-M113V relies on extracting the patient's own T cells, engineering them, and multiplying them. If the patient has progressed far into the disease, if the virus has already decimated their CD4 T cell count, the quality and quantity of the raw material you have to work with is compromised. Early diagnosis ensures you have a robust, healthy starting population for the manufacturing process.
Got it. Okay, second reason. Monitoring viral load, VL. This is the gold standard for success with ART, but how is it used in the context of a cure pathway?
VL testing is the primary measure of efficacy after the intervention. The ultimate success of IMC-M113V isn't whether it reduces viral load while the patient is on ART. ART already does that perfectly well.
The real test is what happens when you stop ART.
Exactly. The measure of success is whether the viral load remains undetectable after the patient has stopped ART. Researchers need to monitor these patients for years post-therapy to ensure the engineered T cells are maintaining that biological control. VL testing is the routine, reliable way to check if the functional cure is holding.
And the third area is where the research gets highly technical, tracking reservoir changes. This moves beyond standard clinical tests, right?
Absolutely. This is where advanced laboratory assays become the engine of discovery. You can't claim IMC-M113V shrinks the reservoir if you can only rely on clinical VL tests, which just measure free-floating virus in the blood. The research itself relies on these sophisticated, specialized, and often custom-designed experimental tests.
Like the QVOA test you mentioned earlier.
Exactly, the quantitative viral outgrowth assay, or other assays that measure the integrated HIV DNA in resting cells.
So those QVOA tests where they take T cells and try to wake up the virus in a lab, that's the actual measure of how many hidden bunkers IMC-M113V successfully destroyed.
Precisely. We need to count the remaining capable viral genomes. These advanced research assays are the only way to scientifically validate the core promise of IMC-M113V. Without this kind of testing, the research dies on the vine. And this also requires specialized genetic testing, specifically to confirm the patient even has the HLA-A02.01 marker to be eligible in the first place.
And finally, there's the simple, non-negotiable role of supporting clinical safety and optimizing this personalized medicine.
T-cell therapies are complex. Every participant undergoes rigorous immune profiling, which is just a fancy name for advanced testing, before, during, and after the infusion. This makes sure the patient is eligible, that their immune status is stable, and that the engineered cells are performing safely without causing dangerous immune overreactions. Testing is the safety net that lets us do this futuristic medicine responsibly.
So the direct message to the listener is that by knowing your status today, you are literally future-proofing your health.
You are. These new breakthroughs only become available to you when you know your HIV status early. Early testing gives you faster access to existing RT, which ensures better immune recovery and sets you up with healthy T cells. And critically, knowing your status makes you eligible for future clinical trials and eventual commercial therapies like IMC-M113V, which are highly contingent on specific genetic and viral baselines. Knowing your status early puts you ahead of the virus, positioned for the most advanced care humanity has ever developed. If we connect this research to the bigger picture, we can start to sketch out a potential ambitious timeline. Researchers are not expecting IMC-M113V to go from a lab bench to a pharmacy shelf overnight. They've mapped out a potential decade-long trajectory for how this technology moves to widespread availability.
And this roadmap is essential because it manages expectations. It underscores that this kind of biological engineering requires patience and rigor. So what are the milestones for the first 3 years? The optimization phase?
So, Years 1-3. Focus on optimization. This is the crucial stage for large-scale clinical validation. We move from small safety studies to expanded, larger Phase 2 and early Phase 3 trials. These will involve more diverse geographic and demographic populations.
And a big part of this phase is about the logistics, right? Making the process itself better.
Exactly. The focus here is on engineering efficiency. How do we make the T-cell manufacturing process faster, cheaper, and more reproducible on a global scale?
And what about optimizing the actual biological therapy during this time?
We need to answer key questions about dosage and specificity. Researchers will be intensely focused on optimizing the cell dose, finding that critical number of engineered T cells required for maximum effect with minimal side effects. Plus, while IMC-M113V targets the HLA-A02.01 GAG peptide, scientists will be busy identifying and validating new immune markers.
Because not everyone has that HLA type.
Right. HIV is diverse. Future versions of this therapy will need to target other viral components or use different HLA markers to broaden its applicability to the entire U.S. population, not just the third who have A02.01.
It sounds like this phase is all about making the treatment more potent, safer, and applicable to the widest possible patient base.
Exactly. Once that optimization is solidified, we can transition to the middle phase. Years 4 to 6. Focus on access and integration. Assuming the trials keep showing strong efficacy and safety, this is the period where the regulatory approval process accelerates and the therapy starts to leave the strict research environment.
What does integration specifically involve during this phase?
We would anticipate the beginning of early access programs for patients who have few alternatives or who are highly motivated, maybe initially for those whose viral reservoirs are resistant to standard RT. And crucially, this is where we would see the integration of the manufacturing infrastructure into major medical centers.
So building out the facilities to actually do this.
Yes, the infrastructure to do apheresis, genetic engineering, and specialized infusion needs to be built. We would also start to see aggressive exploration into combination therapies pairing IMC-M113V with other advanced strategies.
Okay, so we're scaling the manufacturing and delivery. Now for the finish line, the final phase.
Years 7-10. Focus on outcome. This is where the long-term durable data really matures. Researchers will have tracked those early trial participants for close to a decade after their therapy to definitively confirm that they are in durable long-term remission. That's the definition of a functional cure.
And if that long-term data holds up.
If it holds, we would anticipate regulatory approval from the FDA, for instance, for widespread use. And if successful, this phase promises a major societal outcome, a statistically significant reduction in HIV transmission in the U.S. population, driven by the broad availability of this cure-based T-cell therapy.
It's clear this likely won't be a single silver bullet, but rather one powerful weapon used as part of a larger arsenal. IMC-M113V is one important piece of the puzzle.
Exactly. IMC-M113V will likely be integrated into the most powerful, multi-pronged HIV cure strategy humanity has ever built. This multi-layered attack is essential because HIV is complex, it's highly adaptive, and it establishes that deep latency.
So let's talk about those complementary strategies that could amplify IMC-M113V's impact. The most radical one seems to be gene editing tools.
When people hear gene editing, they usually think of CRISPR. And CRISPR-based research is focusing on two main strategies for an HIV cure. The first is a kind of cut-and-paste approach, using CRISPR enzymes to precisely locate and physically excise the integrated HIV genome from the infected cell's DNA.
So you're literally just deleting the virus.
Deleting the virus. So IMC-M113V clears the functional reservoirs, and CRISPR could come in to mechanically eliminate the stubborn traces of DNA left behind.
What's the second strategy?
The second strategy is prevention through modification. It involves genetically modifying the host cell's DNA to make it resistant to future infection in the first place. For example, by knocking out the CCR5 receptor, which is the doorway HIV uses to get into the cell.
So you're improving the resistance of the environment while you clear the infection. That's a phenomenal combination.
It's true synergy.
Okay, what's the second complementary strategy that works alongside this?
Broadly neutralizing antibodies, or BNAbs. These are powerful antibodies, either naturally occurring or engineered, that can attack multiple strains of HIV at the same time. They're typically infused regularly to provide systemic protection.
And how do they help the T cells?
Well, while IMC-M113V is highly specific, targeting that single internal GAG peptide, BNAbs provide broader systemic coverage. They neutralize any circulating virus particles and can help clear out remaining low-level infected cells that the T cells might have missed.
So they act as a general cleanup crew.
A systemic sweep. They prevent new infections or reactivation events, while IMC-M113V goes after the entrenched hidden cells. They cover different stages of the viral life cycle.
And the third strategy is the most preventative, therapeutic vaccines. We mentioned they're different from traditional preventative vaccines.
Right. A therapeutic vaccine is designed not to prevent an initial infection, but to control the virus after an infection or after a curative intervention like IMC-M113V. The goal is to stimulate the patient's native immune system to maintain long-term control after the IMC-M113V therapy has cleared the bulk of the reservoir and the patient has stopped RT.
So the engineered T cells clear the ground and reduce the viral threshold, and then the vaccine makes sure the native immune system is perpetually trained and ready to handle any tiny viral flicker that might emerge later on.
Precisely. Together, these tools, gene editing for removal and resistance, engineered T cells for reservoir destruction, BNAbs for systemic neutralization, and therapeutic vaccines for long-term native immune memory, they represent a truly comprehensive and layered cure strategy based on validated scientific pathways. #outro.
So what does this all mean for us today? For the listener, living in a world where HIV is still a chronic condition, IMC-M113V represents the most promising shift in approach we've seen today. It empowers the immune system. It targets those previously untouchable hidden reservoirs.
It supports the possibility of long-term remission without daily medication.
And it works alongside advanced lab testing to ensure both efficacy and safety.
This therapy is truly one of the most exciting and realistic steps toward ending the HIV epidemic in the United States. It's shifting the status quo from decades of lifelong management to genuine biological control. It signifies a pivotal moment where the functional cure moves from a distant hope to a tangible reality we can plot on a 10-year timeline.
It's an incredible, optimistic leap built on painstaking, highly specific science. We started this deep dive with the premise that an HIV-free future was once considered impossible. And now, thanks to developments like IMC-M113V, it feels absolutely within our grasp.
And this highlights a monumental, overarching shift in modern medical strategy. The cross-pollination of these highly sophisticated cancer-fighting strategies like personalized T-cell engineering into the realm of chronic infectious diseases.
That's a fascinating point.
I mean, think about the implications of engineering immunity for other challenging chronic viral diseases like hepatitis B, or even persistent infections like certain chronic Lyme cases. Where else could this CAR-T and TCR methodology be applied next? The boundary between infectious disease treatment and oncology is collapsing, and that confluence is exactly where the most rapid future breakthroughs across all fields of medicine will occur.
An incredible thought to chew on. But to bring it all back to the present moment and to your immediate health, remember, these new breakthroughs only matter when you are in the system and you know your status early. Everything begins with that foundational test,